Your goal for today is to understand how the ribosomes use the information from DNA to make proteins. Another name for a protein is a polypeptide. We'll talk about that later in this presentation. But first I want to start by going backwards and kind of remind you how we got to where we are. First, remember when it comes to making proteins, the instructions have to come from a DNA molecule which is inside the nucleus. The first step to this process is called transcription. During transcription, RNA polymerase creates an mRNA strand. However, this mRNA strand isn't finished. It needs to be edited or processed before it can be sent out from the nucleus. Remember, we said that when it's processed, the RNA strand is being edited to cut out introns. Introns were commonly referred to as junk DNA. They're kind of losing that terminology these days because we're beginning to understand that they're actually fairly important. We're not really 100% sure of their significance, but they do get processed out because they're not important for making the protein part. And once they're processed out, the mRNA strand is now free to leave the nucleus and head out to the ribosome, and now we can begin the translation process. So let's start looking closer at translation. So this slide right here is a nice synopsis of the total process of translation. It's got all the key players and all the key events are represented here. First, we are out in the cytoplasm at a ribosome, and our messenger RNA has made the trip here from the nucleus. And remember, one of the key things is that anytime we have three nucleotides together, like U, G, G, we call that a codon. And we have a series of codons here. In fact, we have 4 of them. The other player, along with the ribosome and the messenger RNA, is going to be the tRNA molecules, and we have 4 of them here. And two interesting things about the tRNA molecules is that they all have an anticodon at the bottom of them, which is this area right here, and they bring in an amino acid at the top. Now, PHE is not the full name of the amino acid, it's just the abbreviation. Typically, we like to abbreviate amino acids by just using the first 3 letters of the amino acid. PHE is actually short for phenylalanine. But in high school, not incredibly important that you know that. So those are our key players. This area down here, by the way, with the 3 bases at the bottom, the AAG, this is a special area called an anticodon. And let me explain how this all comes together to work to make a polypeptide or a protein. So the first thing that has to happen is the mRNA carries the list for the amino acids in the order that they're to be added. And the way the list is coded is through the codons. So UGG codes for a very specific amino acid. In fact, it codes for tryptophan. So let's take a closer look at how this coding system works. Then we'll come back to this diagram. So let's try to remember UGG before I forget, because I've already did it once. UGG is our mRNA codon. Every codon codes for a specific amino acid. What you're looking at here on the left is a large table of all the possible codons that you could have using the 4 bases found in mRNA. And it also indicates exactly which amino acid would be coded for by that particular codon. You may have noticed that 3 of the codons don't code for amino acids at all. Instead, they code for stop. There's one here, here, and here. And the stop codons are equally as important, if not more important, than some of the amino acids because they're telling the ribosome, hey, you're done, stop. And at that point, the ribosome stops adding peptides or stops adding amino acids, and everything is done. So stop is not an amino acid. It's a command to stop making or stop adding amino acids. So if we go back to our UGG over here, in order to read the codon table, I would have to take a look at our first position, and our first position for our amino acid, for our codon, is the U. So we come over to our first position side, and we find the U row. It's right there. Our second position is a G. So now we know where we have to be looking in this column over here. And so we know we have to be in this box thanks to our U and our G. And we're looking for UGG. It's this one down here. And notice it codes for the tryptophan, which we learned that it should. So each codon codes for a specific amino acid to be brought in. The tRNA molecules are actually bringing in those amino acids, and the way they know that they've got the right thing is that they match up their anticodon areas with the codon. So remember our UGG codon? Look at the tRNA molecule that just brought it. It just brought in the tryptophan, and it had the anticodon of ACC. When we compare ACC to UGG, what we notice is that they're complements. U goes with A, G goes with C, and G goes with C. So the anticodon has to complement the codon. That's how the ribosome knows that it's got the right amino acid to add to the chain. So when we see the next RNA codon down here, which is AAA, the corresponding anticodon would be UUU, because that's what goes with it. So that lets us know that we are bringing in lysine, which is the correct next amino acid. And this continues all the way through. And as they come in, the amino acids are being linked together to form a long chain. Each bond in here between amino acids is called a peptide bond. Hence the term polypeptide. That's why we sometimes call proteins polypeptides, because they're made up of lots of peptide bonds. And the ribosome is going to continue adding more amino acids to the right side of this chain until it reaches a stop codon. And that's how it'll know when to stop. And once it hits the stop codon, it's done. So one of the things that you're going to be able to ask to do is, if I give you a sequence of DNA, you need to be able to tell me what amino acid should be brought in. So if I start with the DNA sequence, T, A, C, which is what most DNA sequences are going to start with when we're making a protein, the first thing you have to do is you have to convert this information into RNA. So we have to transcribe this in our heads. So to transcribe, we just need to know the complements. T goes with A goes with U. Remember, this is RNA, that's why I use the U. C goes with G. So we just transcribed our original DNA sequence into an RNA sequence. So now to figure out what AUG codes for, we go to our codon table here on the left. So our first position is an A, our second position is a U. That tells us we need to be looking down here, and sure enough, there's AUG, and it codes for methionine. So what we just did is we just translated. So we started with TAC, we transcribed it to AUG, and now we know that it's going to bring in the methionine as the amino acid. Again, you don't need to worry about the full name. As long as you know the first three letters, you will be okay. So remember, we started off with DNA, we translated it into an mRNA sequence, and then from the mRNA sequence we were able to determine the amino acid to bring in. One error that students commonly make is they'll try to look at DNA to figure out the amino acid. They'll try to look at the tRNA to figure out the amino acid. That's not what matters. When we're trying to figure out what amino acid we bring in, we need to know the mRNA, because that is what we can use this table for over here. The codon table doesn't work for anything other than the mRNA. So on your worksheet that you have now, you can begin working on translating from the mRNA strands into the amino acids by using the codon table. Sometimes they'll give you the DNA to start with, so you have to shuffle right down through in natural order. Other times you'll have to start with the mRNA, and then you have to go back to DNA, and then forward to the amino acids. Just depends on the particular scenario. The one thing that I haven't covered here is tRNA. Just remember that tRNA has to complement your mRNA. Right? tRNA has to complement your RNA. And you should be good to go. So from here, for the rest of the day, begin working on the worksheets that I passed out yesterday. If you have any problems, feel free to save them. Ooh, I'm sorry, I don't know what happened there. Sorry, Sarah and Reagan. And I will see you on Monday.